Thank you, Daniel. It's my great pleasure to introduce today's speaker, Dr. Hans Jacobs. He's a laboratory specialist in medical immunology at Radboud University Medical Center in Nijmegen, the Netherlands. He has specializations in oncoimmunology, cancer immunotherapy monitoring trials, and biomarker development for in vitro diagnostic applications. He serves as the head of the National Reference Center for Monoclonal Protein Diagnostics at Radboud University Medical Center. He also coordinates the Dutch external quality assessment program for monoclonal protein diagnostics, and he's been doing that since 2012. He's a national board member of the Dutch Foundation for Quality Assessment in Medical Laboratories, known as SKML, for the section on humoral immunology, and national board member for calibrations. He also works within the IFCC framework. He's involved in the harmonization and standardization initiatives for monoclonal protein diagnostics. He's co-authored more than 60 book chapters and primary articles. And since 2016, he's an editorial board member of CCLM, the Clinical Chemistry and Laboratory Medicine. And if he's not too busy doing all that, he finds time to teach at the University of Twente, which I hopefully didn't mispronounce. My Dutch is limited. I'm trying to pronounce some of these locations. But it really is my great pleasure to introduce Hans today, and he's going to talk to you about monoclonal protein diagnostics, in particular aspects with patients who are treating with some of the new biologics that are out on the market. So with that, I'll hand the floor over to Hans, and I will go on mute.
Thank you, Daniel from EFLM, for the invitation, and thank you, Chris, for this kind introduction and also for moderating this session. I know that you logged in from Ottawa, Canada, so it's pretty early for you this morning. And thank you, participants, for all joining this EFLMM webinar. I think it's going to be an interesting session in which we will talk about M-protein diagnostics, and I will demonstrate that the introduction of monoclonal antibody therapy in multiple myeloma patients has introduced new challenges for laboratory specialists in the field of M-protein diagnostics. Let's see if I can now proceed. M-protein diagnostics is performed in the context of monoclonal gammopathies, also referred to as plasma cell dyscrasias. And monoclonal gammopathies are characterized by a monoclonal expansion of plasma cells, mostly in the bone marrow. And in multiple myeloma patients, this expansion interferes with normal hematopoiesis, and patients often suffer from anemia, bleeding, and frequent infections. Bone pain and renal problems also often occur in newly diagnosed patients. Lytic lesions can be seen in classical X-rays or other imaging, and plasma cells observed in bone marrow biopsies are both increased and morphologically abnormal, and cytogenetics can be performed as it has prognostic value. But of course, these monoclonal plasma cells, and that is the topic of today, also produce a monoclonal antibody, and that is secreted in the serum and provides a unique biomarker. Serum proteins can be secreted with, can be screened with protein electrophoresis, which is a technique that separates charged proteins in an electrical field, and this can be done using gel electrophoresis or capillary electrophoresis. In the routine clinical setting of my lab, we make use of gel electrophoresis. So most examples in this presentation will make use of gel electrophoretic techniques. Depending on the charge and the structure of the protein, each protein has a unique migration speed, and each band can be made up of one or more proteins. For example, the big band is caused by albumin. Healthy individuals, and it's good to mention that the band intensity corresponds to its concentration. Healthy individuals, they produce approximately 20,000 polyclonal antibodies each second, and because these antibodies are highly polymorphic, they migrate over a wide area, spanning the entire region entire gamma region, into the beta region, and often even further, and sometimes even further. And that situation is very different for a patient with a monoclonal gammopathy, as these patients produce a monoclonal antibody with one unique charge and a unique mass, and they migrate to one unique spot in the gel, giving rise to an additional band in the spectrum, provided of course that the concentration of that monoclonal band is high enough. In literature, these endogenously produced monoclonal antibodies are called M proteins, but they are also referred to as paraproteins, myeloma proteins, or monoclonal components. So serum protein electrophoresis can be used to screen for these M proteins. And because band intensity corresponds to the concentration, the M protein can also be quantified with densitometry. And finally, with specific antisera against specific antibody components, the M protein can be further characterized in a so-called immunofixation electrophoresis. And then, based on the M protein characteristics, together with other clinical and laboratory information, the monoclonal gammopathy can be further classified in a variety of disease entities, ranging from relatively benign conditions such as MOGUS, up to conditions with high morbidity and mortality such as AL amyloidosis and multiple myeloma. And as this pie chart demonstrates, by far most patients with multiple myeloma have an IgG M protein. Some have an IgA M protein or a light chain multiple myeloma. Other M proteins are uncommon or even extremely rare, such as an IgE M protein. So why would we quantify M proteins? We do that because we want to classify the disease entities such as MOGUS, smoldering multiple myeloma, or multiple myeloma. We want to further stage these different disease entities. But especially also we want to monitor disease activity. We want to follow up on therapy responses. We want to follow up on disease evolution. And as an example in this slide, the therapy response criteria of multiple myeloma patients defined by the International Myeloma Working Group are shown. And without going into further details of these exact criteria, I highlighted those measurements that directly involved M protein diagnostics. And then the crucial role of M protein quantification becomes immediately apparent. So how do we do M protein quantification? The quantification of an M protein relies on the fact that the band intensity directly correlates with the protein concentration. And therefore, the area under the curve of the M spike or the N spike in the scanning densitometry plot, shown here in orange, directly corresponds to its protein concentration. And it can be calculated, provided that the total serum protein concentration is known. Okay, for example, the immunofixation here demonstrates an IgG kappa M protein. The total serum protein concentration is measured in this patient sample at 80 grams per liter. The M spike of this protein is 20% of total serum proteins, and then we can calculate that this IgG kappa M protein of this specific patient is 16 grams per liter. To allow consistent and good monitoring of an individual patient, it is extremely important that each team involved in M protein diagnostics makes good working agreements on how exactly each M protein is spiked and quantified in that specific lab. And external quality assessment programs, they provide an excellent tool to demonstrate variation in M protein quantification between different labs. From our Radboud University Medical Center, we host an EQA program, M Protein Diagnostics, for 75 labs in the Netherlands and in Belgium. And we send around a sample like this. And when we send around a sample like this, with a highly concentrated IgG-kappa M protein with only little polyclonal background, all participants of this EQA are happy and they score great. As shown here, all participants detect this M protein, and the inter-laboratory differences, so the differences between the different laboratories, the VC value is relatively low. But when the concentration of the M protein is lower, some participants fail to detect the M protein, and VC values start to increase. And diagnostics become even more challenging when small M proteins are masked in a high polyclonal background. However, typically an individual patient is of course monitored only in one center, and between lab variation is not that interesting. Personally, I'm more interested in how consistent each individual lab is in the quantification of a certain M protein, so meaning the within lab variation. And we were also able to test this variation within. Well, because we obtained a large plasmapheresis product with a nice IgM-kappa M protein that provided enough material to send out to the 75 participants 5 times over a period of 5 years. And the participants were not aware that this was in fact the exact same sample. And the quantification of this M protein was very consistent over the 5 years, with an average quantification of the M protein of 6.5 grams per liter and a between lab variation of 22%. However, in this setting we were also able to calculate the average within lab variation. And this was 14%. It's lower than the variation between labs, as expected. But what was striking, though, is that we observed an enormous range in how standardized M protein quantification was in the individual labs. Some participants, they quantified this M protein 5 times in exact the same way. For example, a certain lab 6 grams per liter 5 times, or another lab consistently 7 grams per liter. No variation at all. Very good performance. Other labs, though, were poor performers with VC values over 35%, meaning that in daily practice, that this same M protein can be quantified as 4 grams per liter at a certain day, and at 14 grams per liter at another day. And that would be an extremely misleading result for a hematologist who could falsely misinterpret the results as progressive disease instead of stable disease. If we discuss M protein diagnostics, we must also spend a few slides on free light chains. After light chains and heavy chains are assembled as intact antibodies, they are secreted by the plasma cells. However, light chains are produced in excess of heavy chains. It is estimated that light chains are produced in 40% excess over heavy chains. And they are therefore in the as free light chains in quite large quantities. The half-life of free light chains is short because these small proteins are relatively easily filtered by the glomerulus and reabsorbed and metabolized in the proximal tubuli. So when you actually measure free light chains in healthy donors, the polyclonal free light chain concentrations are low, and the free light chain kappa concentration is approximately as high as the free light chain lambda concentration, giving rise to a free light chain ratio that is close to 1. As mentioned previously, it is observed that patients with a monoclonal gammopathy only secrete monoclonal free light chains instead of intact M proteins. And the percentage of patients, multiple myeloma patients, that only secrete these monoclonal free light chains is not small at all. So even up to 15% of newly diagnosed multiple myeloma patients secrete what we call a so-called light chain only multiple myeloma. And these monoclonal free light chains, more often than intact M proteins, cause significant morbidity. For example, because they deposit as free light chain networks in the glomeruli, or because they accumulate in the distal tubuli of the kidney, causing, for example, cast nephropathy. And although these monoclonal free light chains are in fact the world's first cancer biomarker ever, the diagnostics of monoclonal free light chains is often still challenging. And that is because they more often polymerize, giving rise to a smear instead of a clear monoclonal band, and then because they have a short half-life, and therefore they have lower serum concentrations are observed in these patients, even in newly diagnosed patients. In the serum sample pictured here, we do observe a monoclonal band, and the antisera now shown are exclusively reactive with kappa chains. And when we antisera directed at the light chain hidden epitope, where the light chain is anchored to the heavy chain, then we can readily observe in this patient that indeed these kappa light chains are monoclonal free light chain kappas, kappa free light chains. However, in other patients, serum protein electrophoresis is often not sensitive enough to detect additional bands. And for example, if you have clinical suspicion of multiple myeloma or AL amyloidosis, it is crucial to advise for alternative and more sensitive tests to detect light chains, such as Bench-Jones analysis in urine, or measuring serum free light chains, in which an abnormal free light chain ratio is indicative for monoclonal gammopathy. And when we do a serum free light chain analysis on this patient, we indeed observe that without any monoclonal bands detected in this patient, we do observe an abnormal free light chain kappa free light chain ratio with an abnormally increased free light chain kappa. And this patient was diagnosed with a plasmacytoma, localized clonal plasma cells. So, to summarize what we discussed so far on M protein diagnostics, intact M proteins can be monitored using serum electrophoretic methods, in which monoclonal bands are measured. And to detect and monitor monoclonal light chains, alternative and more sensitive free light chain immunoassays can be used, in which an abnormal free light chain ratio indicates monoclonality. M protein diagnostics is performed as screening for monoclonal gammopathies, but it's also used as part of the diagnostic workup and for disease staging. And it's good to realize that the most important role of M protein diagnostics is the monitoring of disease activity. For example, monitoring a possible progression of a MOGUS patient to multiple myeloma, or monitoring a multiple myeloma patient and the treatment responses and disease relapse in these multiple myeloma patients. And these relapse conditions are defined in the IMWG response criteria. So let's shift gears now towards treatment. Treatment of multiple myeloma has substantially changed over the past decade, with the introduction of several classes of new and effective drugs that have really greatly improved the rates and depth of responses in multiple myeloma patients. And one of these novel drugs that are used are monoclonal antibody biologics. And the interesting feature of monoclonal antibodies is that they are highly specific and can therefore be used for targeting therapies. For example, monoclonal antibodies targeted against unique cancer epitopes. And in the early days, the general thought was that for effective anti-cancer therapy, it's crucial to improve the efficacy of antibody-based cancer therapy by conjugating these antibodies to, for example, immunotoxins, radioactive isotopes, or drugs. And it was, however, soon realized by the research community that also naked antibodies, specifically targeted to tumor cells, can indeed cause tumor killing by a variety of mechanisms, such as antibody-dependent cell-mediated cytotoxicity and complement-dependent cytotoxicity. And in the past 2 decades, we have witnessed an enormous expansion of clinically effective biologics. The list shown here is definitely not extensive. That target tumor cells in a variety of ways. They can, for example, directly target the cancer cells, but it has also been effective, for example, to target the tumor vascular system or modulate the immune system of the tumor microenvironment towards increased immune activation. Also for multiple myeloma, 2 biologics have been approved by the FDA and the EMA for administration in multiple myeloma patients. And in a recent review by Tuzo et al., it is shown that novel targets are specifically explored in the context of multiple myeloma, with many interesting candidates currently investigated for therapeutic applications. In a cancer setting, biologics are often administered over a longer period of time, and for optimal efficacy and to prevent immune responses against these biologics, techniques have been developed to convert the monoclonal antibody produced in foreign species into chimeric, humanized, or even fully human monoclonal antibodies. As mentioned previously, daratumumab, targeting CD38, and elotuzumab, targeting SLAM-F7, both directly target multiple myeloma cells, and they are the first 2 biologics approved for the treatment of multiple myeloma patients. In large clinical studies, the clinical benefit in terms of reduction of tumor activity and overall survival was clearly shown for both biologics, without significantly more adverse events, except maybe for the monoclonal antibody associated infusion related reaction that occur in up to 50% of patients. They are mostly grade 1 to 2. In our academic center, we have more experience with the administration of daratumumab. Therefore, in my example in this presentation will focus on daratumumab. Daratumumab is a CD38 targeting monoclonal antibody that has a pleiotropic mechanism of action. And that can be divided into direct effects, Fc-dependent immune effector mechanisms, and in immunomodulatory effects. First, the direct effect of daratumumab, this induced by cross-linking of CD38, causing apoptosis. The Fc-dependent immune effector mechanisms, they include antibody-dependent cellular cytotoxicity or antibody-dependent cellular phagocytosis, complement-dependent cytotoxicity. But it is now shown that daratumumab also has an immunomodulatory effect via the eradication of CD38 positive regulatory T cells, CD38 positive regulatory B cells, and CD38 positive myeloid-derived suppressor cells. And this results in an improved T cell expansion and potentially a better host anti-tumor immune response. To obtain fully human monoclonal antibodies, daratumumab is produced in a mammalian cell line using recombinant DNA technology. And daratumumab is an IgG1 kappa monoclonal antibody, fully human. It is dosed at relatively high concentration, 16 milligrams per kilogram weekly for 8 weeks, and then every 2 weeks for 16 weeks, and then it's dosed every 4 weeks thereafter. Stephen Xu and Pamela Clements and their group at the Janssen Research and Development, they intensely studied the daratumumab pharmacokinetics, and the daratumumab pharmacokinetics are complex, and that is due to its target-mediated drug disposition, which lead to time and concentration-dependent pharmacokinetics. So at the start of therapy, unbound CD38 causes target-mediated clearance of daratumumab binding to its target, and over time, with repeated dosing, tumor burden reduces, and because CD38 positive cells are eliminated and CD38 target saturation can be maintained with lower dosing of the daratumumab. So administration of daratumumab at 16 milligrams per kilogram weekly for 8 weeks rapidly saturates the target-mediated clearance, and dosing every 2 weeks for 16 weeks and every 4 weeks thereafter is sufficient to maintain target saturation. And from their pharmacokinetics data, it can be concluded that daratumumab serum levels can be up to 1 gram per liter in individual patient. And it is realistic to anticipate that these concentrations can be observed with serum protein electrophoretic methods. And what is shown here is our very first patient treated in the Radboud University Medical Center with daratumumab. And the patient was included in a study with an IgG kappa M protein of 20 grams per liter and only little polyclonal background, shown here. During daratumumab treatment, we clearly observed a second monoclonal antibody band, a monoclonal band, that could be characterized as an IgG kappa monoclonal antibody, shown here. And when we ran in the same settings daratumumab spiked in normal cell line, we could observe that daratumumab exactly co-migrates with that second band, from which we could conclude that the additional band was indeed caused by the administration of daratumumab to this patient. And in most patients, that additional band will not interfere with routine diagnostics, especially if the laboratory specialist is aware that daratumumab is administered to the patient. However, we can define a certain group of multiple myeloma patients in which it can interfere with response assessment. Because to reach complete remission or stringent complete remission, the originally monitored M protein may no longer be visible on immunofixation electrophoresis. And in those patients with an IgG kappa M protein that exactly co-migrates with daratumumab, this is a problem. For example, the patient shown here has a high concentrated IgG kappa M protein before therapy, and after several cycles of daratumumab, the monoclonal band has greatly reduced, and at this time I can no longer predict whether the remaining band, IgG kappa band that you see here, is caused by the administration of daratumumab. It may be caused by the endogenous M protein itself, or it is caused by a combination of both. And therefore, in collaboration with Janssen, a Shift Assay was validated that abrogates this interference. And this assay is called the DIRA assay. It's a daratumumab specific immunofixation reflex assay. and it relies on the use of mouse anti-daratumumab monoclonal antibodies that bind daratumumab and therefore alters its migration on immunofixation, shown here. The daratumumab band is shifted more anodal to more anodal migration. And this assay is currently being utilized in clinical trials to distinguish daratumumab from endogenously produced M protein by immunofixation electrophoresis. So in order to be able to confirm complete remission in myeloma patients treated with daratumumab. The procedure needs an additional pre-analytical step in which the patient serum is pre-incubated with the mouse monoclonal anti-DARA antibody, and also the interpretation of this DIRA assay needs experience. But fortunately, this procedure only needs to be performed in a very selected group of multiple myeloma patients treated with daratumumab. And this is what I will explain it's also, this figure comes from a publication from Niels van der Donk from Amsterdam, and he shows that we only need to perform DIRA or any other shift assay analysis in a specific group of patients that have obtained very good partial responses. And if then the IgG kappa, it needs only be performed in patients that started with an IgG kappa, and IgG kappa protein is now reduced towards less than 2 grams per liter. If the urine immunofixation is negative, and if that IgG kappa M protein co-migrates with daratumumab, then it's important to perform a DIRA assay. Because when this DIRA assay is positive, we know that there are still disease activity scored, and it's a very good partial response. But when the DIRA assay is now negative, we can perform other tests and finally conclude if there is a complete remission or stringent complete remission. For example, with what kind of patients then you would perform DIRA testing on, or another shift assay that would be a patient like this. And currently already newer generations of the DIRA assay are available, such as the Hydra shift assay, in which an additional pre-incubation step is no longer needed, and interpretation of the data is also simplified. As different monoclonal antibodies target different pathways, it is very likely that they have a synergistic effect when they would be used in combination. If, however, several monoclonal antibody biologics would be combined in a single patient, that would of course increase the complexity of response assessment using routine techniques. And already alternative, highly sensitive mass spec analysis to monitor M proteins and biologics have been published, including from our own research group, that can be multiplexed and do not disturb from interferences when monoclonal antibodies are combined. It was also investigated whether daratumumab could also interfere with free light chain measurements, but this seems not to be the case. The authors here detected no significant differences in free light chain concentrations in neat serum samples versus samples spiked with daratumumab. And the samples spiked in this paper were spiked at DARA concentrations of 1 gram per liter. As an educational sample, we also prepared a serum sample that was spiked with daratumumab in our Dutch EQA program on M Protein Diagnostics. And we spiked normal serum with 5 grams per liter daratumumab for participating labs to learn how daratumumab behaves in their specific electrophoretic methods. And all participants detected this IgG kappa M monoclonal band. And of course they were not aware that the sample was spiked with DARA, so they named it an endogenous M protein. It was also very consistent with the quantification of this sample, with an average reported M protein concentration of 4.9 grams per liter. We also asked to perform serum-free light chain analysis, and as expected, all participants reported a normal free light chain kappa and a normal free light chain ratio, because daratumumab does not interfere with free light chain testing. To mimic how a more realistic DARA concentration would be handled by the participating labs, we also spiked another sample with 1 gram per liter daratumumab, and this was detected by almost all labs. Because it is very difficult to accurately quantify such small M proteins, many labs do not spike such small M proteins and reported the concentration as below 2 grams per liter. Those labs that do quantify the M monoclonal band tend to overestimate its concentration. On average, they reported a concentration of 1.7 grams per liter. So, can monoclonal antibodies used for other indications also interfere with serum protein electrophoresis? Theoretically, yes, and it's also proven by our moderator of today. Theoretically, yes, but in practice, they often go unnoticed. And in my opinion, there are 3 main reasons why they go unnoticed. And I will demonstrate that with the example of the widely used monoclonal antibody adalimumab, used for rheumatoid arthritis. It goes unnoticed because serum protein electrophoresis is not commonly performed in RA patients, contrary to patients with multiple myeloma. And in addition, the daratumumab concentrations are a lot higher. It's relatively very highly dosed, and other monoclonal antibodies are dosed at a lot lower concentrations, for example 40 milligrams weekly or every 2 weeks. And also important difference between multiple myeloma patients and rheumatoid arthritis patients is that multiple myeloma patients often suffer from hypogammaglobulinemia caused by disease process, and also caused by induced by the therapy that they receive. And RA patients often have a hypergammaglobulinemia caused by the disease process. So it's more difficult to detect small monoclonal bands in a high polyclonal background compared to monoclonal bands with almost no polyclonal background. It is also important to note that CD38 is also expressed at low levels on red blood cells. So therefore daratumumab can bind to CD38 molecules present on reagent or donor red blood cells, and thereby interfere with blood bank compatibility tests, such as false positive indirect antiglobulin tests, and these positive IITs may persist for up to 6 months after cessation of daratumumab therapy. Several strategies can be used to negate daratumumab interference with blood group compatibility testing to ensure that patients receive a timely transfusion when needed. And this includes a chemical method to denature CD38 and reverse binding of daratumumab. Alternatively, binding of daratumumab to red blood cells can be prevented by using neutralizing agents such as anti-idiotype antibody or recombinant soluble CD38. And furthermore, extensive human erythrocyte antigen phenotyping or genotyping can be performed prior to starting CD38 antibody therapy. So these are all things that can be done to perform safe blood transfusions. Because of new therapies and combinations of therapies, including biologics, an increasing number of patients respond to therapy and achieve complete remissions. And this results in an increased survival of these patients. However, we cannot cure this disease, and thus sooner or later, the few remaining cancer cells eventually cause disease relapse. And it is estimated that currently more than 50% of newly diagnosed multiple myeloma patients achieve stringent complete remission. And what is shown here is a schematic representation linking therapy responses to survival. And the figure shows that the deeper the response, the longer the survival. And it also illustrates that current techniques are not sensitive enough for adequate patient follow-up. As a consequence, it is currently impossible to monitor the disease activity in individual patients with stringent complete remission. And there's, because of that, a strong delay in detecting disease relapse. So to solve this problem, there is a lot of research and development with a focus on molecular assays on bone marrow. One of the techniques used is multicolor flow cytometry to detect minimal residual disease in bone marrow. But most techniques for minimal residual disease focus on molecular techniques to detect the rearranged B-cell receptor on clonal plasma cells. And for example, quantitative PCR using allele-specific oligonucleotides or next-generation sequencing is used. However, of course, bone marrow biopsies are not ideal to repeatedly monitor for minimal residual disease. On the one hand, because we know that there is quite some tumor heterogeneity which could lead to sampling error. And on the other hand, bone marrow sampling is an unpleasant procedure for an individual patient. And therefore it is interesting to measure M proteins in blood samples could be an alternative for minimal residual disease testing on bone marrow samples. In general, these mass spec techniques are based on either the unique mass of an M protein, or they are based on unique M protein peptides. And with that, I would like to conclude this webinar. But of course, I am very happy to take any questions if there are any. Let's see how I'm in time.
Perfect. Thank you, Dr. Jacobs. That was a great presentation. I enjoyed that. Clearly, it's a topic of my great interest as well, and I think the participants hopefully found that great as well. So those of you who have questions, I know we have a few here, please type them into the chat box here, and then I'll go through them in order as they show up. So let me go ahead and read the first questions that we have here for our presenter. So Hans, do you know which samples have daratumumab in them?
Yeah, in our institute we find it important. Otherwise, we would be confusing hematologists with second bands or bands that we might or might not report. So, for each newly diagnosed or newly started patient on daratumumab, the hematologist is contacting our lab that specific sample is derived from a patient that has been treated with daratumumab. So then we can also give a better consult, showing that we actually only report the M protein, and we might add an additional comment that the daratumumab monoclonal antibody is seen in this specific patient.
Do you get that information from the pharmacy, or is that in coordination with the clinical hematologists or the group treating?
We receive the information directly from the clinical hematologist, and we are in the group for the transfusion doctors. So they also, of course, both groups are notified that a certain patient is receiving daratumumab, and we log that into our software in which we perform our analysis, so we can interpret that result in the context of daratumumab that is administered.
Do you have a particular comment that you use routinely to indicate what the interference or the presence of daratumumab looks like?
Yes, we have a fixed comment for that, and I can't recall it exactly what we but it's a fixed comment that we invented, and the hematologists are aware that we use that one comment. Yeah.
Okay, so we've got some more questions here. Let's just try and go in order here. So, when do you decide, or who decides when you're going to use the migration shift assay?
It's the decision of the laboratory specialist. So we discuss. So it's very restricted. I don't perform shift assays often. So because there were, as discussed in the presentation, a lot of exclusion criteria. So first of all, the patient should have daratumumab. It should be an IgG kappa M protein that we are monitoring. The IgG kappa M protein should be really low, less than 2 grams per liter. And it should co-migrate on the location that we know is the location where daratumumab would migrate. If all these conditions are there, then it's interesting, especially almost all of these patients are treated as part of clinical trials, and then it's crucial to know is this patient now in stringent. Then we want to have an IFE negative gel, and then we perform this DIRA shift assay or another shift assay.
Okay. Do you use the DIRA assay, or do you use the commercially available Hydra shift assay?
The Hydra shift assay. Yeah.
That makes sense. Okay, we've got more questions rolling in here. Let's go on to the next one. How long after remission do you treat a multiple myeloma patient with daratumumab?
I don't know. This is I don't know that. I would have to dive into the clinical protocols. These are shared with the laboratory specialist. But any questions on the exact dosing and the maintenance and when to I don't how the maintenance therapy exactly looks like. I don't know. I'm sorry.
No, I don't think I could answer that either. The sense I get is that the therapy courses are relatively long, and of course, as you said, everyone on this is basically part of trial data now, so I suspect we'll have more information on that as things go forward. Let's see. Let me ask another one here. What do you see the biggest challenge in reporting serum protein electrophoresis and immunofixation in the context of, let's say, daratumumab and the other biologics which are coming on the market?
Yeah, it's. I think for daratumumab we have now figured out how we can deal with it, how we can circumvent interferences. But if it's going to be combined with other biologics, it's going to be increasingly difficult to have that monitored. So maybe in the far future, or maybe not that far future, it could be alternative techniques. And our group has experiments also with monitoring these biologics, but also multiplexed assays together with the M protein in mass spec analysis. So that could be one way to deal with that in the future.
Yeah, that's good. Do you know if there's many labs using the mass spectrometry techniques to, detect these?
I honestly believe that none of the lab in routine clinical practice use mass spectrometry to monitor M proteins or biologics. I do believe that there is activity. For example, the Mayo Clinic is supporting very interesting data on how they monitor M proteins, making use of the unique mass of these M proteins. But in daily practice, I think it's something for in the future, but not currently practiced.
Yeah, I think that's a good point. So in 10 years, we'll do this again, and you can describe everyone doing this by mass spectrometry instead.
I would be welcome to give a webinar on that in 10 years.
All right. Hopefully the technology gets better for the webinars then too.
I don't see any problems, Chris. I think maybe in the background you're doing fantastic stuff, but I'm just doing my presentation, and it was quite good to do so.
I think it went well. I think Daniel's been helping us in the background a little bit,
Oh,
maybe
very
with
good.
a few people. So we have more questions. This is great. We have lots of them rolling in. Do you have a sense or any experience with the immunosubtraction or immunotyping techniques as far as the biological interferences?
So we don't use the capillary electrophoresis. In general, the serum protein electrophoresis patterns are similar, so the SPE pattern, yeah, you would be able to see this. Also with immunosubtraction, you would be able to see most likely. Again, and I'm not practically familiar with the techniques, but you will mostly also will be able to detect these monoclonal antibodies like daratumumab. If there is also a kind of hydroshift possibility using capillary electrophoresis, I have not heard of so far, so I don't think that is available.
Yeah, I can't say that I'm aware of it either, but theoretically it should still work. I mean, the immunotyping, I think, uses latex particle-bound antibody, so all they'd have to do is bind their anti-sera to the therapy, and arguably it would shift even further than it does with the typical migration shift. But I'm not sure if they're even working on that. I think people end up with gel electrophoresis because it's a little easier to interpret and maybe more sensitive even.
What is happening often is that because the frequency with which you get these samples in which you want to do a shift assay, often these diagnostics are centralized. For example, I know if your patient isn't involved in a clinical trial, there is always a possibility, provided with the PI, that shift assays can be done for individual patients in a centralized way.
Yeah, that makes sense. Okay, so we got another question here. How long does it take for daratumumab to clear from the body? And I guess in the context of how long does it persist before it goes away, let's say by immunofixation or by serum protein electrophoresis?
Now, I believe it will behave like other human IgG antibodies. So let's presume the half-life of also daratumumab will be something like 20 days. And then we can calculate a little bit when we reach the sensitivity of our method. And in general, if you would use immunofixation electrophoresis with a sensitivity to detect intact M proteins of maybe 200 milligrams per liter, then I think at least 1 or 2 months after cessation of therapy, daratumumab could still be visible, especially if you have a low polyclonal background. That is crucial. If you have a high polyclonal background, you will not be easily picking up these small concentrations. Contrary, the transfusion specialists, they seem to have a positive indirect Coombs test even 6 months after cessation of therapy. So they have to be a little bit more patient before they can withdraw all their additional testing.
Oh, yeah, that is persistent. We have another question here for you, and I don't think you talked about it a whole lot because it's not maybe as relevant with an intact, but the question is whether you see daratumumab in urine specimens and by urine protein electrophoresis.
Mm-hmm. In general, I guess not. In general, these intact antibodies are too large to pass the glomeruli, and we will not be able to detect these intact monoclonals, but also the endogenous M protein. However, upon diagnosis of multiple myeloma, we know that many of these patients suffer from renal problems, including problems in the glomeruli. And then it is possible that they start leaking also larger proteins into the urine, and then it is possible, most likely. Actually, I have not specifically looked for this, but then it's theoretically possible, of course, also to be able to detect daratumumab.
Thanks. Okay, I've got one more here for you. I guess there's a few more rolling in. I'll ask this one next. So do you find the clinical users are fully aware of all the interferences? I know there's sort of a black box warning on the drug, and there's been a lot of literature out there. Do you find the users are aware of all the interferences and handling them correctly and understanding it?
I don't know. We are all in our own bubble, of course. I know that especially also the transfusion specialists are very much aware of the potential interferences. I think for M protein a little bit less, or at least reduced to a very specific number of patients, and maybe yes, there is less awareness. So that's why I was very happy to be including a daratumumab spiked normal serum sample to our EQA program, and for some of these labs really showing for the first time that daratumumab is actually visible on immunofixation electrophoresis, and for them to learn where exactly this daratumumab migrates, so they are able to better recognize that in the future.
Yeah, that's smart. I like that program. I should talk to, you actually know one of my colleagues who does the EQA for Canada here, or at least for the province, and I think we could probably do the same sort of thing here. It might be interesting. I've got one more for you here, and the question is, does the HydraShift assay, does it shift 100% of the band? So let's say you had a 1 gram band in the migrate shift. Is there any residual band, or does it all shift out of the way?
Yeah. I know that, and actually we together we have looked at these validation of and can we actually shift all the daratumumab in all of these patients. And actually the width of an M protein is very large, from 0 to up to 50 grams per liter. But the range of daratumumab is very restricted. So we know that daratumumab, in fact, never outreaches more than 1.2 grams per liter. So the reagents that are used are very tightly fixed on that range, and we know for sure that with these shift assays, all the remaining daratumumab has been shifted away from the interesting area that you want to look at.
Thank you. I think that's most of the time we have for questions. It's now the top of the hour, whatever time that is, wherever you are. Maybe that's 3 o'clock where you are, 9 o'clock here. I'd really like to thank you for your presentation today. I enjoyed it very much, and I'd like to thank everyone for attending and all the thoughtful questions. I'll give you a chance for any parting words or advice for people who are dealing with this.
Final words, but I would like to thank you for moderating this session, and also for Daniel, yes, for fixing our software to me, making this possible. And thank you very much for joining this webinar.
Thank you. Thanks, everybody.